Unknown presentation Klebsiella pneumoniae by Shelby Sanders. What is the significance of IDing specific organisms? According to a lab manual provided by Dr. Hurst, stated that the role of clinical laboratory in a hospital is to quickly and efficiently identify the causative agent of a patient's infection. The experiments that I performed was the litmus milk, gram stain, carbohydrate fermentation, nutrient agar citrate, and urase. Methods and materials for gram stain. I inoculated the specimen on a slide and allowed the specimen on the slide to air dry. Then I heat fixed the slide by passing it through the Bunsen burner 3 passes. I flood the slide with crystal violet dye and let the dye sit for 1 minute, then rinse dye off with water. I flood the slide with gram iodine, then let the dye sit for 1 minute, then rinse the slide off with water. I rinse slide with ethanol until the slide runs clear, then rinse with water. Flood slide with saffron red for 45 seconds, then rinse dye off with water. Then I blot dry slide with bibulous paper, and then I observe slide utilizing microscope using oil immersion lens. Gram stain was the first test that I did. Gram stain allows me to visualize whether or not my specimen is gram positive or gram negative. Also allowed me to see the morphology of the specimen. This test showed me a pink rod or gram-negative rod. The picture that I utilized for this slide was from Google because I couldn't take a picture utilizing my camera, so I just used a picture from Google that looks very similar to what I saw in the microscope. This canceled out the possibilities of the specimen being a cocci and my specimen being gram-positive. This test crossed out the possibility of my specimen being Staphylococcus aureus, Lactococcus lactis, Micrococcus luteus, Corneobacterium, and Bacillus cereus. The second test that I utilized was the litmus milk test. Litmus milk let me know if my specimen is acidic or alkaline by it fermenting lactose. Also, if my specimen is acidic, does it produce a curd? My results showed a pink liquid in the test tube with whitish curding sticking on the test tube. My results eliminated the possibility of A. faecalis, Pseudomonas aeruginosa, Salmonella, and Proteus virigus being my specimen. So this left E. coli, E. aerogenes, and Klebsiella pneumoniae. The third test that I did was the carbohydrate fermentation. This test determines if the specimen eats the 3 sugars lactose, dextrose, and sucrose. Once the specimen eats that sugar, it produces acidic waste, turning the test tube liquid yellow. If the specimen produces a gas, during this process, it produces a bubble in the Durham tube. In all 3 test tubes with lactose, dextrose, and sucrose was yellow with a bubble in the test tube. This test led me to believe that my specimen is E. aeruginans, Klebsiella pneumoniae, or the possibility of E. coli. This is the fourth test that I performed. This test helps us distinguish between different types of pathogenic bacteria found in the stomach by looking at the ability of an organism to use nutrient sodium citrate as a sole carbon source. If the slant remains green, this means the specimen tested negative for citrate. If the specimen is positive for citrate, the slant will turn royal blue. My results prove that my specimen is positive for citrate because of the royal blue color. This test eliminated the possibility of the specimen being E. coli. This led me to believe that my specimen is E. aeruginans and Klebsiella pneumoniae. This is the fifth test that I performed. This is the test that determines if the specimen is either Enterobacter or the Klebsiella. Positive results for urase turns the liquid in the test tube pink. Negative results stays unchanged or turns into a pink yellow color. Normally, Klebsiella has positive results and Enterobacter has negative results, but a young lady in the microbiology class as me and I determined through our test tubes that we either had Kleb or Entero, but both of our tests came out negative for urase. This led me to look at one more test to determine what my specimen is. The agar slant. Agar slants help us visualize cultural characteristics while observing both of our specimens. characteristics. While observing both of our specimens, her specimen had a thick, white, and glistening growth. My specimen had a slimy, translucent, and raised growth. This proved that my specimen is Klebsiella pneumoniae. Why is this specimen important to identify? Because it causes UTIs, it causes pneumonia, it causes meningitis, it causes bloodstream infection, and it also causes pyogenic liver abscesses. These are my results, and that concludes my presentation.